Am I audible?
Yes, Archana, you are audible. Can you please introduce, sir?
Yes, sir. Yes, ma'am. I can introduce. Uh,
Okay.
First of all, a very good evening to everyone. A warm welcome to the session 13, where we will have a lecture on the topic of hidden features of the human hippocampus and its clinical significance. Now, let me give the brief introduction about our lecturer. Dr. Anand here is presently working as a research associate in the Imaging Research Center, Department of Radiology, Cincinnati Children's Hospital Medical Center, Cincinnati. Since August 2022, he is closely working with MR physicists, pediatric clinicians, and radiologists towards developing accelerated MRI acquisition method to rapidly scan awake pediatric population. Before moving to the Cincinnati, he was working in the University of Alabama at Birmingham along with the neurologist in epilepsy division and helping him in the post processing of high resolution MR images. Particularly, he and his PI were actively investigating the role of hippocampal dentation and its influence in the memory performance of healthy controls and epileptic patient. Prior to UAB, he was working for Philips Healthcare India Limited as MR and research and development consultant for the 19th month at Fortis Memorial Research Institute, Gurgaon, where he was working on T1 perfusion imaging in gliorama grading. Dr. Anand obtained his PhD degree from Anna University. His current area of research interest include T1 perfusion imaging for breast cancer diagnosis, gliolama grading, quantifying hippocampal dentation in epilepsy patient, and developing accelerated MRI acquisition method. He has co-authored 18 peer-reviewed journals and 22 international conferences. He has actively collaboration with researchers from IIT Madras, IIT Delhi, IIT BHU, and VIT Vellore. He is also an editorial board member of BMC Medical Imaging. Now, I would like to say that, sir, you can start your lecture. Thank you, sir.
Thank you, madam. Thank you very much for the wonderful introduction. So let me share my slides. Can you see my screen now?
Yes, sir, we are seeing your screen.
Okay, thank you. Okay. I think someone needs to mute. Okay, thanks. So today, let me try to introduce some of the hidden features of human hippocampus and its clinical significance. So before doing that, let me try to throw some light on what is hippocampus, what is the hidden feature that I'm talking about, because basically, so this is basically some kind of MR related or MRI related topic, but you can hear me, right? Everything is fine, right? I just want to double check. Can you see my slides moving?
Yes,
So
sir.
no, so we
But we can see your Gmail email,
Okay.
but unable to see the PPT.
Okay. Give me a second. Sorry about that. That's what I was thinking. Okay. How about now?
Yes, sir, now it's visible. You can go for the
Okay.
slide mode.
I think I'm in slide. Can you see my slides? I'm just changing the slides. Can you see that in real time?
No, sir.
But you can see my first slide,
Yes,
right?
sir.
Hidden
Yes, sir.
features.
First slide is visible, but it
How about this one?
no,
The
sir. Only
second
first
slide.
slide is there. Sir, you can go for the slide mode. Now it's changing.
Sir, can you reshare your screen and selecting on
Okay.
entire screen mode?
Okay. Let me. I don't know what's going on. Give me a second. So what do you see now? Like my slides or okay. How about now? Is it visible? Can you see my slides moving?
Yes, sir, it's visible, and I would yes, sir, it's moving. It's moving.
Okay, thank you. Thanks for the information. Like usually I use my Teams app, but this time I try to use the browser, but I have a lot of doubts. So I'm going to talk about a hidden feature that was not well investigated in the research field. That's called hippocampal dentation. So our group uh, was actively investigating this dentation thing, and we were the first group to report uh, first paper on this dentation. So this is something related to anatomic. Also, we have developed some imaging methods like high resolution images or some upsampling schemes to capture this feature. But one problem with this is that these features are not readily seen in any hippocampus. Like these are kind of hidden, meaning we need high resolution images to capture these features. So that's why the title stands like hidden features of human hippocampus, meaning like the features are available, but it's not readily visible. So that is the whole idea of this particular talk. So this is how I want to introduce so most of this research and all these papers were published when I was for UAB, University of Alabama at Birmingham. So this is our beautiful green campus of UAB. This is the scanner that we used, Siemens scanner, 3 Tesla magnet, Siemens magnet. So we had a research facility called Civitan International Neuroimaging Laboratory. That's a kind of a separate division or a separate group that coordinates and organizes all the research activities related to MRI. And these are our technicians, these two guys here. So, and this is our epilepsy division. So epilepsy falls under the Department of Neurology. So basically that's a hospital with a medical college, which offers UG, PG medical programs. So he's the PI, so the one who is tall here, and he was our division director. So all these are our epilepsy group. Okay. So most of the slides, most of the results in these slides are from these two papers that we published. One is in epilepsia, which is more related to a clinical domain, like is a clinical journal. And this is human brain mapping, which is kind of usually people report methods that they have developed on MRI or any other imaging modality related to brain. So we published our findings in these two journals. So that's why I have included this in the slides. Okay, some basics about MRI. So we basically know from basic clinical sciences or basic engineering science, like whenever we disturb well-established magnetic field, some sort of current is produced in the coil, right? Solenoid or whatever it is. So this is a basic principle that works behind the MRI. So when we put, so basically when we enter the MR machine or when we enter the MRI area, which means that as far as you move close to towards the magnet, all the proton or water molecules in the human body or tissue will get strongly influenced by this magnetic field, which means that the magnet is always on. So that's the whole idea. So when we put the subject or a volunteer participant on the scanner, the tissue protons, water molecules, align with the existing magnetic field. So which means that it is obeying the magnetic field around its the environment. So at that moment, we are applying a RF pulse or a waveform, a pulse to excite that, so that the water molecules, the proton molecules, the protons will undergo precession, meaning trying to rotate in different axes towards the magnetic field to attain the resonance. So once it attains the resonance, it's going to it's going to undergo the precision with the same resonance with respect to the existing magnetic field, or we can call that as equilibrium state, right? So the moment we remove the RF pulse, it's going to lose its energy and undergoes relaxation, which there are two types of relaxation called longitudinal and transverse relaxation. So these are the two relaxation processes that takes place, which means like it is going to decay exponentially. So in that case, it's going to emit some energy, which is going to get captured by a coil behind that, which is called receiver coil. Sometimes we have coils called transmit and receive coil, meaning the coil can send the RF pulses as well as it can receive the signal from the due to this relaxation process. So that particular signal is going to get detected by the coil, and which is going to generate some kind of current, right? So that's called the NMR signal, which means that nuclear magnetic resonance signal. So basically in the early times, it was called as NMR and not as MRI. So that's why the traditional name stands still, like nuclear magnetic resonance signal. So this is basically an electric signal. So there comes kind of analog to digital conversion, and then we get the raw data, meaning the raw data is something which is going to be very noisy and it is not going to be informative, which is just the collection of information based on the application and removal of RF pulses and disturbing the magnetic field. So once we have built this raw data matrix, then we apply Fourier transform kind of transformation, which transforms the raw data into a meaningful image. So that is why here comes the Fourier transform. So this is also called reconstruction. So all these process like application of RF pulse and reception of NMR signal falls under the field of pulse sequence programming, and application of Fourier transform and generating the image from the raw data is called reconstruction. So these are two important field in the area of MRI. So this is the basic idea. So just I want to give some basics about this. Otherwise, I think most of us would have got MRI done. So I had a lot of volunteer. I have volunteered for many studies. Like hopefully, like I should have done at least some 50 plus MR scans. So meaning like I had volunteered for the scans. So it is totally non-invasive. It is not harmful as X-rays or CT because no radiation. Just we are playing with the magnetic field, right? So it's totally safe. Okay, and based on this principle, there are many sequences available, meaning variety of scans available, starting using different imaging principles or methods called spin echo, gradient echo, inversion recovery, and whatnot. Meaning like the pattern in which we are going to apply the RF pulse and the excitation, and the way we receive the signal determines the kind of sequence. For example, spin echo, which means that we will apply two RF pulses, one 90-degree pulse. After that, we'll apply 180-degree pulse. So by doing this, we are getting as much as information about the body tissue, and we want to very, very efficiently use that raw data to generate weighted images that are going to be very, very distinct with respect to different, which is going to be very, very distinct based on the tissue component. For example, in the brain, we generally say white matter, gray matter, cerebrospinal fluid, right? So fluid is more of water, which means that there should be more population of protons or hydrogen atoms, right? So this particular population is going to vary with respect to white matter and gray matter. So we want to efficiently represent that in the matrix form. So that's why we have got T1 weighted, T2 weighted, proton density weighted, which means that is going to give you some image based on the density of water molecules available in that particular voxel or tissue. And gradient echo, T2 star, inversion recovery, which means that before applying the RF pulse, we will just invert the magnetic field by applying a 180-degree pulse, which means that this is going to give you a long time for the molecules to relax. So you can get a lot of RF signal or the NMR signal because the relaxation time has been expanded now. So that is the basic idea. And diffusion weighted is like based on the movement of water molecules, and perfusion weighted is like, this is very popular in tumor imaging tumor patients, like particularly like glioma patients, grade 1, grade 2, or grade 3, whatever it is. Like perfusion, which means that when you have some kind of tumor developed on the tissue, it's going to affect the BBB, brain, what do you call that? Barrier disruption. So which means that it is going to cause some insult on the tissue, which will allow the perfusion of contrast that is injected during the scan. So based on that, you will see a lot of enhancement, signal enhancement on the MR images, using which the radiologist can take a better decision of the treatment strategies for the patient. And this is functional MRI. This is totally different. I have not done anything in this field. And susceptibility weighted, of course, you should be exposed to this term called angiography, like more related to cardiology, where you have a dye applied and try to see the kind of the obstruction or whatever it is in the veins. And this is SWI, which is going to again exploit some properties of magnetic field, and based on which they do a lot of work related to tumor grading or whatever it is. So these are the sequences But in our study or in our today's presentation, like we have used only T1 weighted images, and this slide is particularly like just to give you some idea about existing MR sequences. Okay, different views of human brain. So we are just trying to get into this topic today. So this is the human brain. So basically, this is T1 weighted MR image. So basically, we have three different views. One is sagittal view, axial view, and coronal view. Sagittal view is like when you see a person from the side views, left or right. So this is called sagittal view. So when you see someone from the top, that's called axial view. And when you see someone face to face, and it's going to be coronal view. So axial view is like something like slicing the tomato or whatever it is. Okay. And this is how the axial view will be observed in three different MR sequences, like T1 weighted, T2 weighted, proton density weighted, flyer weighted, T2 star weighted. So if you take the T1 weighted images, whatever you see in white are called white matter, and whatever you see in gray color are called gray matter. And whatever you see in dark are called cerebrospinal fluid. So this is how we want to distinctly differentiate three different tissue components of human brain. To the opposite, if you see that the same image in the T2 weighted, it's going to be in opposite, like the white matter is going to be in gray color and gray matter is going to be some kind of white color, and water is going to get very well enhanced and highlighted in the T2 weighted images. So that's why you see water to be more bright compared to other tissues. So basically, we are trying to use these properties based on the water molecules to diagnose the disease condition very effectively. For example, if someone gets hit or meet with an accident, like his brain, this is going to got some kind of brain injury, right? That is one possibility. Another case, like due to aging, you will see a lot of loss of neurons in the human brain, which is going to affect the shape changes of brain structures. So in that case, we will use T1 weighted images. And for that, when you want to read brain tumor in a patient, basically brain tumor is like it's going to have a lot of heterogeneous component, meaning like we have got a lot of components of the tumor, glioma, like enhancing component, non-enhancing component, and what do you call this? Water molecules, like due to inflation and like recurrence, sometimes recurrence of disease, or which is called like, I don't remember that name exactly, like loss of tissues, or we can say like dead cells. Okay, so all these are based on water component. So we use T2 weighted in that particular case, and flyer is sometimes again for tumors where you see a lot of. So when you have a non-enhancing tumor, meaning the application of contrast will not be useful there. So in that case, we use flyer. So basically what I want to say is like all these sequences are meant for different disease conditions, treating different kind of patients. But everything is based on three different tissue components like white matter, gray matter, and CSF. See, we always want to develop sequences to efficiently differentiate three different components so that a radiologist or neurologist can effectively make his decision to treat the patient. So there are four different lobes in the brain, starting from frontal lobe, parietal lobe, occipital, and temporal lobe. So basically, every lobe has got its own functioning. So our topic, hippocampus, falls under this temporal lobe, which is temporal lobe is meant for memory component of healthy individuals or human subjects. So frontal lobe is primarily for to think and make decision or whatever it is. Occipital lobe is controlling our vision activities. So let's talk about more about this temporal lobe here. Okay, so this is a whole big picture about hippocampus with other brain subanatomic structures. So what you see here is the hippocampus. You see some kind of undulation here. So in addition, before hippocampus is like amygdala. This is a different region. We are not going to touch any of these regions like hypothalamus or basal ganglia, thalamus, amygdala. We will talk only about hippocampus today. So every region in the brain has got different functions to control and govern, meaning, for example, hypothalamus will take care of the body temperature, hunger, fatigueness, or sleep. So when our sleep gets disturbed, which means there's something wrong with this hypothalamus or it's not signaling properly or giving instructions properly. So basal ganglia is some kind of region that takes care of body movements, learning habits, cognition, emotion, whatever it is. And thalamus is also responsible for regulation of sleep, consciousness, alertness, and hippocampus is primarily meant for memory performance of individual. So when you see some kids or when you see some professors are very, very sharp and very active and totally outstanding, which means that naturally their hippocampus should be something different from any other hippocampus. So that is a basic idea. So that is the main root cause or the main reason for investigating this hippocampus in our research papers. Okay, where exactly is hippocampus in the human brain? So this is how you see hippocampus here. So basically we have got two hippocampus or hippocampi, left hippocampus and right hippocampus. So for example, if someone is left-handed individual, is going to be right hemisphere dominant. So his right hippocampus will be very, very active. And if someone is right-handed individual, his left hemisphere is going to be dominant. So his left hippocampus will be very, very dominant. It's going to take the major control over his memory performance. So this is the basic idea that we need to understand. So in right-handed individuals, his left hippocampus is going to be very active and vice versa. Okay. Okay. So let's get into the topic. So whatever I'm showing here, these images 1, 2, 3, 4 are T1 weighted MR images, just focusing hippocampus. So what I want to talk today is, you see here like some kind of a weird structure here, or I can say some bumps, or some kind of speed breakers, or some kind of ridges, or we can say like tooth-like appearance, something similar to AXA, right? So this is the kind of feature which is not readily seen on any images. We need high resolution images, or we need to upsample the images to the high resolution space to visually see and appreciate this feature. So this is called hippocampal dentation. So these are dentees. So what is the basic idea, or why do you want to care about this? The reason for that is that this particular region or this particular inferior surface is composed of specific cells in the hippocampus, CA1 and subiculum. So these are the different regions in the hippocampus. These two different regions are specifically dedicated to memory performance of individual. So when you see here, like when you see a lot of bumps here, which means that the neurons can communicate each other very, very quickly, or the firing can happen very, very quickly. That is going to reflect in its memory activity or memory performance or cognition. So when it is flat, something like this, a neuron from this region will take a lot of time to communicate to another neuron in this region. Okay, so which means that either his memory should be may not be outstanding. I don't say it's going to be very bad, when it is bad, which means that he is not a normal healthy human being, meaning he is going to be a subject, a disease subject. So this particular feature has got a lot of things to do with memory performance. So that's what we are trying to understand here. So the hippocampus is a complex brain structure in the temporal lobe. So basically this falls in the temporal lobe, and dentation is a feature of hippocampal morphology that looks like convolutions or bumps in these regions called CA1 and subiculum. And this is the inferior aspect of hippocampus, and this is the superior aspect of hippocampus. Okay. And this particular feature is not readily seen. That is one thing. The other thing is like that varies dramatically, even in the healthy individuals. And it is positively correlated with memory performance. So my PI has developed a high resolution sequence, T2 weighted high resolution sequence called HR micra, to image this hippocampus in the high resolution space so that you can see the dentation here. So he correlated dentation of healthy individuals with their memory performance and saw that there is a strong correlation between dentation and memory performance. But the problem is he did the investigation using subjective method, meaning he manually counted the number of dentees here. For example, 1, 2, 3, 4, 5, 6, 7, 8. So the dentation count of this particular hippocampus is 8. And here in this case, it's like 1, 2, 3, 4. So basically he counted this to be 4. So subjectively he counted this and he published the first paper. The problem is like this cannot be done or repeated by any other doctors or any other researchers who want to pursue hippocampus or dentation in their research studies. So to facilitate that, we developed an objective method, meaning using computers and programs to quantify this dentation and see how well this quantification is by doing some correlation analysis between objective method and subjective method, and see if there is a correlation, then what is the correlation with the memory performance. So all these are the basic studies that we did. Okay, so this particular slide is from the basic paper that he published for the first time. So if you see here, the left, the first one, the first plot here. So on the x-axis, we have got left hippocampal dentation rating, meaning he used a rating scale to subjectively quantify dentation. So the dentation score is from 20 to, 0 to 8 here. And there are some memory performance tests the individuals underwent, meaning California Verbal Learning Test 2 acquisition T-score, and we have got a few more like ray delayed recall, ray recognition. So basically these are verbal and visual memory scores to test the performance of hippocampus. So he found that left hippocampal dentation rating showed a statistically significant positive correlation with CVLT, California Verbal Learning Score and long delay free recall. So this means that these are right-handed individuals, but their left hippocampus showed positive correlation with memory performance of their memory performance. Whereas we don't see any correlation with the right hippocampal dentation. Okay, so the idea here is like to show that dentation has got something to do with the memory performance of individuals. So later we will apply this particular concept to temporal lobe epilepsy patients or patients suffering from memory related disorders. Okay, so that is the whole idea behind this. Okay, so we had high resolution scans to visualize hippocampus. What if the research group doesn't have that facility, they have only standard T1 weighted images? So should we drop the research or how to proceed further? So these are the questions. So we developed a high resolution pipeline to use standard T1 weighted images to see dentation. Okay, we will discuss this. Before that, let's talk about these points here. So dentation, though you can see dentation here. So for example, figure A is the cropped zoom in version of hippocampus where you see dentation here. So if you want to quantify dentation, the first step is like you have to segment this hippocampus. Okay. So unfortunately, when you use any tool to segment hippocampus in the native space, which means that standard images are acquired at 1 mm resolution. Okay. So when you do this, the segmentation results also will be in 1 mm. So it cannot effectively represent the undulations in the segmentation. So you don't see dentation effectively, or you hardly see 1 or 2 dentes with no prominence. So that's what this figure is showing here. Rather, when you upsample the images to a super resolution space, the segmentation that you do here on the hippocampus will pay due respect to the undulations here. And when you transform this segmentation results into 3D surface rendering, you will see dentation showing up here. So that's how we have showed that in arrows. Okay. So, but this is not true with a hippocampus which is naturally flat, or there is no dentation at all. It is it's not going to exaggerate any results. So there is no dentation at all here. Okay. So what is the reason behind that? The reason is that the voxel size, which is 1 mm, in the native resolution is relatively large compared to the curvature of the dentated surface, which meaning the undulations that you see here is very, very small compared to the voxel size. So this cannot capture that effectively. So what to do? How to handle this? You have to zoom in your camera so that that can effectively capture. What do you mean by zooming? Which means that you are going to upsample or resample. So the fine scale contour information of the hippocampal boundary is lost when a grayscale image is reduced to a binary segmentation. Okay. So we have developed a method which I'll show that in the next slide, how to resample or upsample, and then we can effectively capture this. So idea is like your voxel resolution has to be very high, meaning 1 mm is very low. You have to push the resolution from 1 mm to 0.5 mm, 0.5 or 0.3 mm or 0.225, so that you can zoom in your resolution, zoom in the image to effectively capture these fine structural changes in the high resolution space, so that you can effectively do segmentation and get your beautiful 3D surface. Okay, so what is the methodology? So the methodology, what we followed here is like we developed this subjective method and applied this to 59 temporal epilepsy patients. So there is a term called hippocampal sclerosis, which means that when healthy hippocampus is fine, in the case of disease condition, you will see neuronal loss happening on the tissue, right? So which will dramatically affect this dentation. So which will result in loss of dentation, loss of hippocampal volume, which is called as hippocampal sclerosis. Okay, so another point is like some of our participants virtually had no dentation, even in the normal hippocampi. So that is true. And we used Philips Achieva 3D scanner, and all our images were acquired in the native resolution of 1 mm, which is going to be, which we will treat to push it to high resolution space. So we developed a segmentation approach where we upsample the data from 1 mm to 0.25 mm isotropic, which means that it is going, the voxel size is high. The low voxel size of 1 mm is now is going to be 0.25 mm, meaning like you see a lot of zooming in effect in the voxel size to see what is exactly going on this particular specific voxel. Okay, and since you upsample the data, the file size is getting big and big. So we reduced the file size by cropping just the region of hippocampus and getting rid of other unwanted brain structures. And we used a software called ASHES software. So I want to mention one point here is like this particular software, ASHES, Automatic Segmentation of Hippocampal Subfields, is a freely available software. Anyone can download and do the hippocampus segmentation. So if someone of you is pursuing a PhD and want to do something, want to try something with this hippocampus stuff, you can definitely go ahead and use this. And you have a lot of images available in public domain database. So you can make use of this. Okay. And okay, so we use this as a software to do our segmentation. And we also use, so I will also show some basic block diagrams of this software tool. And there is another software called ITK Snap, which is existing for a long time. You can use binary segmentation results to generate 3D surface rendering. This is also a freeware, you can make use of it. And okay, so before doing that, we visually score the dentists in all our subjects, which means that that is a subjective way. So whenever you develop some objective method, that needs to get validated with the benchmark, right? So that's a subjective visual score. Okay. Okay. This is the kind of ultra high resolution pipeline that we develop to do segmentation. The first step is we take the T1 weighted MP range standard images. So MP range is magnetization prepared rapid acquisition gradient echo. So that is basically the sequence name that is used here. Naturally, that is the that is basically a scan type or a method used. So it need not be MP range all the time. It can be any standard 3D T1 weighted images. That is the requirement. And you use another free software tool called FSL Toolbox. That is a command called FLIP. We use spline interpolation in that particular toolbox to resample the data. So most of the tool softwares used in most of our papers were from freewares, freely available softwares, because doctors always want to do research using freely available methods or technologies so that anyone can repeat the study, anyone can get benefit out of it. So if you write some MATLAB code or if you use some Python or even Python is okay, but any sophisticated programs, softwares that needs a lot of training or that cannot be replicated in a new environment or whatever it is, so that is going to cause challenges. So research wants to be open to all, and results should also be open to all so that anyone can make use of it. So that is the basic idea. Okay, so we crop the images from, we have sampled the images from 1 mm to 0.25 mm and just we crop the images and we fed the images, 2 input images to this ASHES software. One is like the original full T1 weighted 1 mm isotropic images. The other one is 0.25 mm images. So they need this tool, particularly this tool with PEN TLE whole hippocampus ASHES atlas needs 2 images, 2 inputs to do the segmentation process. And we get this in figure C, you see this segmentation result here, right? So this is how the results are. And then we used ITK Snap to visualize the segmentation results in 3D surface rendering. So when you generate 3D surface mesh, this is how your hippocampus would look like. And if you zoom in and see, instead of voxels, everything is now in faces and vertices. So each triangle is a face composed of 3 different vertices. So this is how you change this. So idea is like instead of working in the voxel space or domain, we change that to 3D domain where you have a lot of free softwares to handle, treat, manipulate these surface meshes. So that's the whole idea. Okay. And this is a basic block diagram of ASHES segmentation pipeline. So you need, first thing is you need the ASHES software installed, and then you need to download a specific atlas to serve your purpose of segmentation. So in this case, we used PEN-TLE whole hippocampus atlas that will do segmentation of hippocampus on the 3 T1 weighted images. So that needs 2 input. One is T1 and another one is T2. So in this case, instead of T2, we are using upsample T1 weighted images as our T2 input. And this is basically meant for hippocampal subfield segmentation. But in our case, we have used whole hippocampal segmentation. So don't worry about this, but this is how the tool works. So basically, Professor Yushkwich developed this method in 2006. So it's very popularly available and readily available. It's a very popular method. You can make use of this. And here comes the quantification process. So this is the flow diagram that represents the steps in the process. So the first step is you have this surface mesh available, but you cannot use that as such. You need some kind of smoothing to be done so that the pixelation effect or the side effects or after effects due to zoom in, zoom out needs to be treated. So we use a smoothing process, and then we extracted a small portion of the whole hippocampus representing dentation, which we call as swath, so that the quantification could be more accurate. And then there is another step called drawing strip on the swath. I will show you that pictorially to give you a good feel of what we are doing exactly. And then we do some kind of simplification process, and then we quantify area under the curve of the strip to quantify dentation. So basically the idea is like you do segmentation of hippocampus, you transform this segmentation results into 3D surface rendering, and you extract a portion of dentation and draw a line to connect the peaks of the dentines, and that line is extracted and simplified, and you calculate area under the curve for that specific line. So this measurement will represent dentation, and it showed correlation with subjective method also. Okay, next step. So this is how your smoothing works. So all this smoothing, all these steps were done using a free software called Mesh Lab. You can download and make use of it. It's a wonderful software that does a lot of operations on the 3D surface rendering. It helps you calculate surface area, perimeter, and a lot of filters are available. You can play with that. So we do top-in smoothing. We apply top-in filter on our mesh. So figure A is the raw 3D surface rendering, and figure B is the smooth version of it. You see, right? The pixelation effect or ripples were removed, and the surface appears to be more smooth. So the parameters are very simple: lambda, mu, and smoothing steps. We use 0.5 and negative 0.51 and 500 for these 3 different parameters. So you can play with this. And this is obtaining an open cut surface or swath from the smooth surface mesh. So figure A is the whole hippocampus. So we don't want this whole hippocampus. We manually drew or highlighted this portion of dentation, and we extracted that is shown in figure B. And then we did some kind of reorientation so that the undulations that you see here should project in the Z direction, and the plane has to be in the XY direction. So all our quantification will be on the values coming from the Z direction and not from X and Y. So that is why we reoriented that in the XY plane. Okay. And then in this step, we drew or traced a strip on the inferior surface connecting the peaks of dentation, which is shown in figure A. And this is the output of figure B. So figure A and figure B are dentated hippocampus, and figure C and figure D are non-dentated hippocampus or flat hippocampus where you don't see dentation. So which is just to give you some kind of differences between dentated and non-dentated hippocampus. Okay, so some examples of strips viewed from the side. So dentated subjects, you see figure A and figure B, you see here like it looks like some kind of speed bumps or humps in figure A and figure B, and figure C and figure D, we don't see such prominence here. So there is no dentation, but it is curved in nature. Okay, so we are not worried about this curved nature of hippocampus. We consider only this dentation here. And this is the simplification process. So we conceptualized an idea and we transformed that idea into a MATLAB code that does the simplification process of the strip. Because, see, for example, this is figure A just straight away from the mesh lab, and this is figure B. This is a point cloud data. If you have the toolbox, what is the name of the toolbox? Computational toolbox, I think. So that gives you the function called PC read that can capture any surface, 3D surface rendering, and will give you point cloud data. So if you see the zoom in view here, you see here, right, blue dots. So it is not a single line, whereas it is populated. So we need to simplify this. We applied our algorithm and obtained the simplified data here. So the simplified data is shown in this star, right? So circles represent the original point cloud data and star represents the simplified point cloud data. So if your data is simplified, then it is very, very easy to calculate area under the curve. So sorry. So this is how your simplified point cloud data is visualized in the XZ direction. So still, though the strip got simplified, it does not affect the undulations. That is the point I want to mention here. So basically, the idea is you have a complex strip with a lot of values, but that is not simplified. So we want to skeletonize or simplify the strip so that you can effectively quantify. Okay. So conversion of curvilinear line into straight line using segment length calculation. So basically, if you see here, I said this is a flat hippocampus, but it is curved in nature, right? So this curvature portion may slightly corrupt our AUC measurement or objective measurement. So we want to make it straight without affecting dentation. So to do that, we calculated segmental length and appropriately altered that, modified the strip so that that is going to be straight in line, but still preserves segmentation. So if you see figure A, this is actual data. You see it is curved in nature with some dentation. Figure B is, there is no Y direction, just segmental length and Z direction. So XY has been modified into segmental length S. So which means that it is straight line data, but still that has dentation. So figure A and figure C represents 3D curvilinear data. Figure B and figure D represents 2D straight line data, but still has dentation. Now comes the actual part in the objective method, which is area under the curve calculation. So basically, when you see some kind of undulation, and we want to quantify that, the basic step is just to filter the data and subtract the results from the original data to see the difference, right? So that is the same basic principle we used here. So in MATLAB, we used a function called Savitsky-Golay low pass filter to quantify this undulation here. So figure A is the original data and figure B is the low pass data. And figure C is just the overlay of original as well as the filter data to show you some difference. So if you see here, this is the difference data. Figure D is the difference data. So whatever undulations you see here are the difference that resulted from the subtraction of filter data from the original data. And we calculated area under the curve for this difference data that is shown here. And figure F is the truncated data, which means that there are some points that we want to discard from in the beginning as well as in the end. So that is, we just truncated the data to get the actual effect of dentation. So the AUC here for this particular case is 9.03 units. So this is how we did the quantification. So we applied this particular process on all the hippocampus to get the AUC measure from every hippocampus in all the subjects. So we also visually validated this AUC measurement to see really that makes sense or not. So what we did was like we arranged the hippocampus left or right, whatever it is, from highly bumpy or prominently dentated hippocampus to flat hippocampus, and see how this AUC number varies across these hippocampi. So say for example, figure A, if you see here, it is prominently dentated with lot of dentis here, and the AUC is 12.53. Whereas if you see here, figure F, it is a flat hippocampus, and the AUC is just 2.15. So which makes it makes sense. Like the AUC is varying with respect to what we see visually. So this is the kind of initial validation we did, and to make sure that everything is in good shape. Okay. Now comes the results and discussion here. The first step, the first result that we got is like the novel segmentation approach, the high resolution approach that we developed demonstrates dentation clearly across all the subjects than native resolution segmentation. So that is the first result that we find. And one more problem with this is like, though it is an objective method, still some manual steps are involved in this. So to answer this question, what we did was like we hired two raters, meaning two individuals, to draw the swath, draw the strip, to see how differently these subjects do the stripping process and see what is the variation. So we conducted intra and inter-rater study, and we calculated the ICC, that's a coefficient, which was found to be greater than 0.99. So we repeated that 3 different times across 2 different raters, and we found that the results were outstanding. Meaning, what is the idea? What is the point here is that this particular process of drawing swath and drawing strips and simplifying is a very, very simple step that can be taught to any individual, and anyone can adopt this method, and anyone can effectively do this process without any problem. That is the idea behind this. Okay. And the third result is like the AUC values were found to be significantly different between epileptic and contralateral hippocampi, and significantly correlated with visual counterabilities. So what does it mean? So we applied this process on temporal lobe epilepsy patients. So to understand this result, we need to have some background about temporal lobe epilepsy. So it's a disease called epilepsy, which means that so whenever we think or whatever it is, like our neurons in the brain will undergo a process called firing. That is how it communicates with other neuron to process information or help you to control other body organs or whatever it is. So firing happens. So when this firing happens very, very rapidly due to a disease condition, then the subject will start developing seizures. Okay. So this particular process is called, when that happens in the temporal lobe, that is called temporal lobe epilepsy. When that happens in the left hippocampus region, then we call it as, then that is going to cause some damage to the hippocampus. So you classify, since you have 2 hippocampus, left and right, we classify the patients into left temporal lobe epilepsy patients, right temporal lobe epilepsy patients. When you say left TLE, which means that the left hippocampus is bad one, or it is undergoing some kind of damage. When you say right TLE patient, which means that right hippocampus is bad and it is going some kind of damage. Okay. So we want to see, we already published this dentation in the healthy controls, and we want to see how this dentation as well as our AUC method works or operates on epilepsy patients. So that is the idea. So what we found was very interesting. Like you see this table 1, statistical analysis of AUC values for ipsilateral and contralateral hippocampi. So we want to understand this term ipsilateral and contralateral. Ipsilateral means when you say ipsilateral side of hippocampus, which means that that is a bad hippocampus. That is the hippocampus from where the seizures are coming out. Contralateral is something a good or opposite. For example, in the temporal lobe, left temporal lobe epilepsy subjects, left hippocampus is the ipsilateral one and right hippocampus is the contralateral one. Whereas in the right temporal lobe epilepsy patients, right hippocampus is the ipsilateral one and the left is the contralateral. So idea is like when you say ipsilateral and contralateral without saying left temporal lobe epilepsy or right temporal lobe epilepsy patient, which means that you're trying to differentiate good hippocampus from the bad hippocampus. So that is the idea. So here, if you see table 1, the AEC values were found to be different for ipsilateral and contralateral. For example, in the ipsilateral case, the values were 4.36, and in the contralateral side, it is 5.67, which means that the AEC measurement is able to differentiate ipsilateral hippocampi from contralateral hippocampi. This is a very important finding. This is a basic step in the analysis. So this is first success. And second thing, table 2, we also grouped the subjects into left temporal lobe epilepsy patients and right temporal lobe epilepsy patients. And if you see here in the left TLE patients, since their left hippocampus is bad, the AUC is less because it is going to, the hippocampus is bad. The seizures are caused a lot of damage to the left hippocampus, resulting in loss of neurons and tissue, volume, dentation. So that's why the AUC is less here, 4.32. Whereas in the right hippocampus, since the left one is not functioning well, some of the activities done by the left were shared by right, and the dentation is much better on the right side, which means that when you have two hands, when you feel something is not okay with the left hand, you keep doing additional works using your right hand. Similarly, when your left hippocampus is not taking part in your memory activities very effectively, right hippocampus will share some load and will try to compensate that particular loss. So that's a basic idea. So that is getting reflected in this measurement. So that's why the values are 5.25. And in the right TLE patients, their right hippocampus is a bad hippocampus. So the AUC is just 4.40. Sorry, this particular logo is disturbing. In the left hippocampus, the values are 6.13. So the idea is like this AUC measurement is very, very effective in differentiating bad hippocampus from good hippocampus as a whole, as well as specifically with respect to left temporal lobe epilepsy patients and right temporal lobe epilepsy patients. So these are the significant findings from this particular study. And also we correlated statistically, we ran some statistics on these numbers to make sure how well it correlates with the subjective measurement. So when you correlate all the, when you do the correlation on all the subjects, all the hippocampi, you got a very good result, meaning for totally we had 118 hippocampi, and the results were statistically significant, p less than 0.0001, which is really very good. And when you do the same correlation on the ipsilateral hippocampi and contralateral hippocampi, still it holds good. It is statistically significant. So meaning that the whole idea is like the method that we developed showed strong positive correlation with subjective method, which means that you need not visually score all the hippocampus across all the subjects. Instead, you can use your computers and replicate this method to quantify dentation. So that is the whole idea from this particular slide. We also applied our, so in the normal healthy individuals, the subjective method showed positive correlation with memory performance. So we tried to do the same here using an objective method. So if you see here, the AUC of we don't see a big statistical significance here on the right hippocampus, but we do see strong significance, a trend towards significance, but it is not statistically significant. So among the left temporal lobe epilepsy patients, California Verbal Learning 2 trials 1 to 5. This is a test, memory score test, which reflects efficiency of verbal memory acquisition, showed a positive trend with AUC values of right hippocampal dentation, but was not statistically significant. So this is basically left TLE patient group where left hippocampus is bad and right hippocampus is good, which means that the right hippocampus is taking part in the memory activity of those subjects, which means that there should be more dentation on the right side compared to the left side. So the AUC method tried to show some kind of trend. If so, the p value is like 0.07. If it is like 0.04, then which means that that is statistically significant. So this showed kind of trend, but it is not statistically significant. So conclusion. So this is the first objective method that we developed that showed strong positive correlations with visual assessment of dentation. The point is like the objective method showed positive correlation with subjective method. And since that is one manual step in the process, we conducted intra-rater reliability test, and we found that ICC was 0.99, which is really good. And we also demonstrated that AUC, dentation, is asymmetrically less in hippocampal sclerosis, implying AUC reflects pathologic changes in the hippocampus. So when you see AUC to be high, which means that the hippocampus is bumpy or highly dentated, and vice versa. So when you see a flat hippocampus in the disease subject, which means that the loss of dentation is caused due to epilepsy. So this is reflecting the pathological changes. Okay, and the last point is a non-significant trend suggests that prominent dentation on the right may be associated with preserved memory function in the left HS, hippocampal sclerosis subjects, supporting the functional reserve hypothesis and possible reorganization of verbal memory function. So this is a big statement which needs a lot of future studies. So if anyone is pursuing your PhD or wants to pursue PhD, you can take this point and you can start your investigation. So the whole story is you have a patient. You have a patient, say for example, left temporal lobe epilepsy patients. Okay. So before he gets into epilepsy disease, he was a normal subject. Okay. This is intervention 1 or time point 1. And after some time, he started developing seizures. So he becomes a left hippocampal, left TLE patient. So which means that his left hippocampus is bad now. So previously, both the hippocampus were good. When we did the imaging, we do see dentation on both left and right hippocampus. Okay. Since he becomes a patient, the seizures are coming from left hippocampus. So he started developing the disease and loss of tissue or neuron, loss of neurons. So volume reduction and loss of dentation on the left hippocampus. This is the second scan that we did at time point 2. Okay. Over a period of time, we did another scan to see how hippocampus looks like. So now, interestingly, what happens is left hippocampus started losing dentation, whereas his right hippocampus started developing dentation, which means that the right hippocampus is trying to compensate the loss caused by the left hippocampus in terms of memory performance, which we call as functional reserve and possible reorganization of memory performance. So When something is not working fine, the other thing is taking care of that, which is resulted in terms of uh, uh, structural morphological changes. Okay, so this particular hypothesis or theme is not yet investigated. So I'm giving you a research problem. So you can start working on this if anyone is interested. If you need any further support, you can contact me. I'd be happy to help you or guide you. So this is the important conclusion we found from this analysis. Okay, and these results were published in, as I mentioned previously, like epilepsy as well as human brain mapping. So epilepsy is all about like the subjective analysis, and human brain mapping method is objective method that we discussed. These are some of our references. And currently I work for Cincinnati Children's Hospital Medical Center. This is one of the best and top rated first in the nation. So when something is first, then naturally that gives you a lot of credit and that invites a lot of funding, right? So we have a lot of funding, and so we have for research alone, we have 3 Tesla machines and 1.5 Tesla machine. So just for research activity, just like working to electronics lab and do some experiments. Apart from that, we have, I think, I'm not sure, but I think we have 11 dedicated clinical scanners to treat patients, to image patients. So this is the kind of facility that we have. And we do have a lot of postdoctoral opportunities for PhDs. So you have completed your PhD recently and you want to pursue some kind of postdoctoral training in MR related activities or MR physics or something, then you're welcome to apply to any postdoc positions in our hospital. You can contact our HR. Okay. So with this, I conclude my presentation. Thank you very much for the opportunity. And if you have any questions, I'll try to answer. Thank you.
Participant, do you have any questions? You can ask.
Thank you, sir. Thank you very much for your nice talk. Really, it will be a very nice session, and especially that of, though it is the last, it will be of very practical oriented experience. That means you shared things. Thank you very much, sir.
Thank you. It's my pleasure.
May I request the participant and the distinguished speaker to turn on the camera to get captured for this session?
Oh, sure. Nice to see Professor Vaithiyanathan here after some long time.
Thank you, sir. Thank you. Sorry, that time I'm on the way actually. Okay, I'll talk to you. Just I got it off your WhatsApp number. Is keep on touch with the mail only. Okay, so surely that hereafter we'll keep in touch actually.
Sure. Thank you so much.
Now we will soon be sharing the exam link for the test and followed by the feedback forms.
Thank
the
you,
text
Anand.
will.
Thank you, sir. Anand, sir. Thank you. And I'll rest of the things I'll keep in touch with you. Okay.
Okay.
So we are proceeding for the next thing. And thank you. Take care, and we'll keep in touch. Thank you, sir.
Thank you, sir. Let me sign off. Okay. See you all. Thanks.